The topic of this talk is multiple views of a dream and what we learned from them. Our team includes members from Helsinki Institute for Information Technology, HEAT, at Aalto University and University of Helsinki, and members from Institute for Molecular Medicine, Finland, SIM, at University of Helsinki. We participated in both drug sensitivity sub-challenges, and this talk is about our solution to sub-challenge 1. Like the other teams, I presume, we were motivated and fascinated by this core task of personalized cancer medicine that humans dream to solve. Given genomic information of a cell, can we predict from it how the cell responds to drugs? Specifically, we were interested in the following 3 questions. A, how can we optimally use the multitude of measurement data which give us different views on the cell and the active cellular processes? B, how can we bring in prior biological knowledge and see how can we learn a model from comparatively few training examples? In a nutshell, our approach works as follows. First, we use several strategies to enhance the existing measurement data, which we call views in machine learning terminology. So for that, we combine different measurement data, and we use gene sets and key genes. So this addresses the first 2 concerns we had about using different measurement data and using prior knowledge. And then we apply a novel multitask, multiple kernel learning method that learns a regression from the data views to the drug sensitivity. And as the author of this method, Mehmet, is he himself, I will leave it to him to explain this part. So, in brief, this multiple kernel component means that we learn from all the data views, and the multitask aspect means that we learn for all the drugs simultaneously. This allows us to deal with the fact that we have quite few training examples available. Furthermore, to avoid overfitting, we make the most important method choices on an external data set, namely Sanger data for drug sensitivity, published earlier this year. And once we train the model, we use it to predict drug sensitivities of all the cells. So in the first step, we construct additional enhancing data views, and basically we considered 4 different categories: measurement combination views, gene set views, focused views, and discretized views. And in total, this gave us 31 data views in addition to the 6 measurement types we have. So the first category we introduce are measurement combination views. And here the idea is that if we combine evidence of multiple different data sources like gene expression and copy number data and DNA methylation, we get a much stronger, much more robust or more reliable signal about the gene activity than if we use the individual data sources alone. Basically, we followed 2 different approaches. On the one hand, a very simplistic one, where we take for each gene a product of different measurement values from the different sources. And on the other hand, we take the gene activity scores from the paradigm data integration tool that was already mentioned earlier today. Secondly, we consider gene set views, which aim at seeing the forest for the trees, so to speak. So here we aggregate values of functionally related genes to obtain robust activity signals at a higher biological level, namely the level of pathways or functional gene sets. And we use two very simple statistics to summarize the gene values, mean or max, depending on the data type. And we used here two collections of gene sets from MSIC database. The third category are focused views, where we exploit prior knowledge on key genes associated with breast cancer. So the needles that have already been identified in the haystack. What we do here that we take 40 genes, so we focus those, we create new data views that contain only 40 genes which have been identified by Sanger Institute as having driver mutations in breast cancer. Finally, we consider discretized views. So the DREAM organizers already provided a binarized version of RNA-seq data, which are supposed to tell whether a gene is expressed or not. And similarly, we produced a binarized version of exome-seq data, which should indicate whether a gene has a stable mutation or not. And to define a mutation as stable, the threshold we said is that the fraction of the alternative allele is at least 0.5. And we discard entries from low quality reads. So this multiple kernel learning method gives us an importance weight for each individual data view. And the result we see here is that all the data views are important. So what the bars indicate are the weights of the individual views, and the horizontal line is the weight we expect with uniform distribution. And we can see that even the view with the smallest weight, so this weight is closer to the expected one than to 0. Here I show the list of the top most important views, and together they contribute more than 20% of the total view weight. And what we can see is that all the enhancing views we newly introduced are among those top 5. And furthermore, exome-seq data in their binary version are most prevalent, so they appear in the top 4 views. On the other hand, the least important views are quantitative exome-seq views. So that means the quantitative exome-seq data do not add much information after we have seen the binarized version. This plot shows again the individual view weights, but now colored by the category. And what we see here is that each category has at least one good view. So this horizontal line is again the expected uniform weight. And the original quantitative measurement data are these pink bars here, and among those the array-based expression has the highest weight. And so what we can see furthermore is that the discretized views and many of the gene set views perform or have larger weight than expected. Finally, we did some analysis about the importance of individual pathways. So in essence, we did a leave one pathway out sensitivity analysis. And the KEC pathways that were most important are shown here. And this seems reasonable in the sense that those contain genes that are relevant for breast cancer. And in future work, it would be interesting to check the pathway importances for individual drugs. This is especially interesting when we know what the drugs are, so when we know the ground truth. And now I give the word to Mehmet.
Hi. Elisabeth already explained the results that we got. Now I will talk about the methodology that we followed. And first, we identified the key challenges from the data provided. We identified 3 key challenges. These are, we have to make use of different information sources. And also, we have a very limited data. We have to learn a reasonable model from this limited data. And the other one is we have missing genomic measurements and also missing drug sensitivities, which means we have missing data on the input side and also on the output side. And our strategy was a principled combination of 4 key components. And we have developed a novel probabilistic algorithm to combine these key components. And these key components are nonlinear regression, multi-view learning, multitask learning, and on top of these 3, Bayesian inference algorithm. And I will explain them one by one. The first one is nonlinear regression. The basic idea is to model the output, model the response as a nonlinear function of the input data. And this approach has 2 implications. First one, rather than using a linear model, using a nonlinear model allows us to decrease the number of model parameters, because the data views provided are very high dimensional, and using linear algorithms means to increase number of model parameters. And the second one is, of course, as you can imagine, the relationship between genomic measurements and drug sensitivities are a highly complex function, and in order to capture this complex relationship, we need some nonlinearity. And in order to achieve this nonlinearity, we converted these views and also enhanced views into kernel matrices, and we basically used Gaussian kernel for real-valued data and Jacquard similarity coefficient for binary valued data. And the second component was multi-view learning, and as Elisabeth already explained, we have 37 views in total, and we have to extract meaningful information from all of them simultaneously. And we followed a particular multi-view learning approach, which is called multiple kernel learning. Basically, we calculated different kernels on top of each view, and we combined them in a principled manner. The third component was multitask learning. As challenge organizers and data providers explained, we have 53 cell lines and 31 drugs, and these are very limited in terms of training data, and in order to get rid of this limited data problem, we have to apply this multitask learning idea. Multitask learning idea basically tries to make use of available data by sharing some of the model parameters between different tasks. For this particular challenge, the task would be each different drug. And for each drug, we identified this regression problem as a separate task but instead of learning them by one, we learned them at the same time. And the last component was Bayesian inference. Basically, it considers all model parameters as random variables, and we learn them. We learn their probability distributions using a approximation, variational approximation in our case. And briefly, I would like to mention the method that we proposed. If you are familiar with graphical models, this is the graphical model that we used. And these plates means replications, and each circle is a random variable, and these shaded nodes are input data. For the views, we converted them into kernels, and for drag responses, these are our output responses, basically. And this first part is the nonlinear regression part. Basically, from each view, we converted these views into some scores using standard kernel-based regression. And in the second part, we combined these view-specific outputs using some weights in order to get the target response. And if you consider multitask learning, as you can see, these weights of the different views are not task-specific. It means that we use these weights for all of the drugs at the same time. And this is the graphical model in its full detail. We have prior distribution for our model parameters, and also these are the probabilistic assumptions that we made. We, of course, have normal distributions for most of these model parameters, and for prior distributions, we have gamma distributions. And at the end, we have developed a variational approximation algorithm that learns all of these model parameters at the same time. Okay, in summary, we applied this new algorithm to these challenge data with these two specific details. We discard drugs with almost constant sensitivities because they don't add any information in terms of prediction. And also for each drug, we didn't try to capture these missing drug sensitivities. And as I already mentioned, nonlinear regression captures the hidden relationship between genomic measurements and drug sensitivities, measured drug sensitivities. And also our multi-view approach gives relative importances of input views. Now we can identify which views are important in terms of prediction. And the last bullet is about limited data size. Both multitask learning and Bayesian inference gives us a chance to get rid of this small sample size problem. Okay, to conclude, in a nutshell again, we have these original data views. We enhance these data views using data combination or prior information. And then we applied our novel probabilistic algorithm to learn from all data views for all drugs at the same time. And we converted these predicted sensitivities into rankings. That's it. Thank you.
I think there's time for a few questions, if there are any. Okay.
So, what kind of interpretability these models allow you to make with respect to the biology?
Okay. Basically, we assumed that the predicted drug sensitivities is a weighted combination of predictions produced from each view separately. And we have used basically a weighted sum of these predictions. And as Elisabeth already showed in this figure, for each view we have a weight, and of course in this case we have a significantly positive weight for all of the views, which means all of them carry some kind of information for prediction. But we cannot say these ones are not important, but we can easily say that this is very much important than the others in terms of prediction.
So if you hadn't had the Sanger data, how different would would the results have been? How much did this rely on training on that?
Okay. Actually, for the Sanger data, we didn't use the data for training. Actually, we used this as a separate challenge, as a separate prediction task. And actually, we have developed 3 different algorithms for the same challenge. And we identified our best algorithm on Sanger data. And we applied this algorithm to the challenge data.
Yeah, I had maybe a question related to the interpretability. At the end of the day, roughly what is the dimensionality of the feature space you're doing the prediction in? Is it
Okay, maybe I didn't mention this explicitly, but if you look at the probabilistic model, we converted all of the views into kernel matrices. And these kernel matrices have the dimensionality of number of cell lines, which is quite small. Compared to using linear algorithms, in that case, this would be the number of genes or number of features that is available for these genomic measurements. But in our case, the dimensionality is the number of cell lines, which is quite low.
So if you had many, many more cell lines, I mean, I guess what I'm getting at is there some kind of biological natural fit for the dimensionality? Obviously, if you had thousands of cell lines, you probably could still do a pretty good job with a much lower number of effective features.
Yes, and also if you have too much available data for your prediction, you can of course use this Bayesian methodology and get rid of some of them using sparsity. In our case, we didn't enforce sparsity because we already had very limited data and we don't want to discard some of them. But if you have many, many cell lines, you can put some sparsity prior and get rid of some non-informative ones.
Hi. So I was interested in the kernels that you're using. So are you learning hyperparameters for the kernels? And if you are, did you try learning? Because you can also have kernels which would allow you to learn which genes are actually important within a specific view. So did you look at kernels like that as well?
Okay, for the kernels, for the binary valued data, we don't have any parameter because we have used Jacquard similarity coefficient. This doesn't have any parameter. And for the Gaussian kernel, we used a simple heuristics, and we didn't play with the hyperparameter values. And regarding the gene importances or pathway importances, there is no easy way to do this analysis because we have used a nonlinear method. But you can, of course, do some kind of leave one gene out or leave one pathway out analysis and train the model and see the performance.
Yeah, I was thinking there are these automatic relevance determination kernels, which then you get back having a parameter for every gene, but then maybe you don't have enough data.
Yes, yes. You can integrate that kind of prior into the probabilistic model, and you can also learn this hyperparameter, of course, but this will increase the number of parameters.
Okay, thank you. Let's